Good morning, ladies and gentlemen. My name is Thipwimon Chompookham. I'm currently studying at Surinta University, Thailand. Today, I'm here to talk to you about ensemble methods with deep convolutional neural networks for plant sleep recognition. To start with our describe introduction and contribution, then I'll mention the framework of the proposed ensemble CNNs, ensemble methods, and plant sleep dataset. After that, we'll consider experimental results, and finally, I'll summarize my presentation. Plants are essential to human life and can be used as food and even medicine. There is a wide diversity of plants in nature. Importantly, some plants leaves look very similar. It is quite difficult for people who are not familiar with the plants to identify them. Thus, the identification of plants requires expertise, such as that of taxonomic botanists and plant scientists. Usually, plants can be classified from various components called plant organs, such as leaves, flowers, bark, seeds, and stems. This researcher proposed artificial intelligence system to stop disease spread and recognize the disease types. Researcher of plant sleep and disease recognition system. Contributions. We propose the Ensemble Convolutional Neural Network method to overcome challenges in plant sleep and plant sleep disease recognition. To discover the best CNN model, we first can find CNN models. Second, we chose the best 3 and 5 CNN models. Finally, the CNN output probability of each CNN model were when given to the ensemble method to do the actual classification. We compare 3 ensemble methods for the plant sleep and plant sleep disease recognition. And this paper also provides the new standard mobile leaf dataset for comparison of image recognition methods. The framework of the proposed ensemble convolutional neural networks. The effect of multiple CNN models on the ensemble learning framework is regularly better than the single CNN model because ensemble learning can well integrate the advantages of multiple CNN models. We introduced the Ensemble Convolutional Neural Network framework to address the plant sleep recognition problems as shown in this figure. The first part of the framework combines with state-of-the-art CNN architectures called multiple CNN in order to find the baseline CNN models. 5 P10 CNN models consisting of MobileNet V1, MobileNet V2, NextNet Mobile, DenseNet 121, and exception. Subsequently, the transfer learning and data augmentation techniques are applied in this step. Second, we chose the best 3 and 5 CNN models called 3 Ensemble CNNs and 5 Ensemble CNNs. Finally, the output probability of the CNN models are given to be recognized by the ensemble methods. We propose to use 3 ensemble methods, namely the unweighted majority vote, unweighted average, and weighted average to do the actual classification. Ensemble method can the sum of weight value are calculated around equation. Plant sleep datasets. We have evaluated these ensemble CNN methods is the mobile leaf dataset and 2-leaf disease datasets. The mobile leaf dataset is collection of 10 cultivars that are taken in the nature environments using DSLR cameras and smartphones. We collected the data from 3 regions of Thailand. The mobile field area are shown in this figure. In this dataset, the mobile leaf images are diverse in brightness, shadow, and even camera angle. Because the mobile leaf images were captured from the nature environment as shown in this figure. Each dataset contains 5,262 images in total. The plants millet dataset is collection of plants images proposed by Penn State University. that correct variants, plant leaf and plant leaf disease. In our study, we select only tomato and corn leaf disease datasets. The detail of this dataset is as follows. Experimental setup and results. The best parameter for each CNN model are shown in this table. We evaluate the single CNN model using 5-fold correlation on the Mobley dataset to avoid overfitting. From the result in this table, it can be seen that DNet 121 outperformed other CNN methods with a correlation accuracy of 98.61%. Moreover, MobileNet V2 is the best CNN model when applied to the test set. The accuracy of the CNN model was slightly decreased by approximately 7% on the independent test set. In this table, we show the experimental result with the data augmentation technique and CNN models on the Mobley dataset. We compare 6 data augmentation techniques. The experiment showed that the best performance is the exception model when training the model with 3 data augmentation techniques with the accuracy of 91.77%. We divide 90% of data at a training set and 10% at a test set. The test accuracy is shown in this table. It can be seen that Kumar and Wani proposed VGG16 to recognition the tomato leaf disease dataset and receive the accuracy of 99.25 without applying the data augmentation technique. In this experiment, the results show that the DNet 121 and exception using data augmentation surpassed or CNN model with an accuracy of 99.87%. As seen in this table, it shows that exception combined with mixed data augmentation technique provide an accuracy of 99.22%. CNN architecture still show the better result than the previous study with approximately 10% accuracy. In this experiment, the outputs after applying the softmax function of every single CNN model are then used in the decision layer of Ensemble method. We use 3 Ensemble methods to recognize the plant leaf datasets. As seen in this table, the accuracy increased by 2.98% on the Mobley leaf dataset. The weighted average Ensemble approach also obtained accuracy of 99.93% and 99.47% on the tomato and corn leaf disease dataset, respectively. The result leads them to conclude that the Ensemble methods can increase the performance of the CNN architectures. Conclusion: in this paper, we have proposed Ensemble Convolutional Neural Network architecture to improve recognition performance on the plant leaf dataset. We evaluate 5 CNN models on the Mobley leaf dataset and 2 plant leaf disease datasets. Second, we select the 3 best CNN models to establish. In this paper, we have proposed Ensemble Convolutional Neural Network architectures to improve recognition performance on the plant leaf dataset. Exception: MobileNet V2 and DNet 121. Call 3 Ensemble CNNs. Additional 5 CNN models are applied at 5 Ensemble CNNs. Finally, 3 Ensemble methods are proposed to classify the output of CNN models. As for the Ensemble CNN, the experimental results show that the 3 Ensemble CNN achieve the highest accuracy performance on tomato and corn leaf disease datasets. Moreover, 5 Ensemble CNNs outperform 3 Ensemble CNNs only on the Mobley leaf dataset. Surprisingly, Ensemble CNN without data augmentation technique obtained the highest accuracy on 2 plant leaf datasets, Mobley and corn. However, more than 99% accuracy was obtained from the tomato and corn leaf disease dataset. In future work, we plan to work on other data augmentation techniques such as generative adversarial networks, auto-augment, and sample pairing methods. Another direction for future work is designing new Ensemble CNN, as well as bio-inspired algorithms such as an artificial bee colony and ant colony optimization will be employed to optimize the weight of the Ensemble method. Thank you for your attention.